Hello students, welcome to the next video. So today we will learn regarding the vitamin B12, that B coenzyme. It is called vitamin B12 coenzyme. Okay, so if you see the structure of this vitamin B12 coenzyme, which is also called as cyanocobalamine. Okay, so you can see that cobalt is been surrounded octahedrally by 4 nitrogen of the pyrrole ring. Okay, like this with a conjugation. Okay, uh, like this. Okay, and you can see here, so one CH bond is here, CH bond is here, but here you can see there is no CH bond directly. It is attached from this here, this to here, like this. There is no CH bond here, so that is the difference between the porphyrin ring. Okay, so this is called Corin ring. This is called as a Corin ring. Okay, so porphyrin ring means it will have here CH bond like we have seen in hemoglobin. Okay, so this is called Corin ring, where one CH bond like this, okay, like this is been missing. Rest is correct. Same pyrrole ring is there, and fifth is coordinated by R here. So that is why it's the best organometallic compound, where metal and R, any carbon, it is attached here. The first organometallic vitamin B12 coenzyme here, biological in the biological system, and the sixth coordinated with 5,6 dimethyl benzimidazole, 5,6 dimethyl benzimidazole, and this becomes a coenzyme. When it is attached with this, we call it as a vitamin B12 coenzyme, and this is a Corin ring, and CONH2 group is attached here. Okay, for the pyrrole ring here, and you can see here this ring is very big ring with having the phosphate group here P, and sugar as well as a base. Sugar will be there and base will be there, so it's a very complicated structure. So in which the cobalt is in +3 state here. Okay, octahedral structure, and surrounded by 4 pyrrole nitrogen of the pyrrole here, and it is a low spin state. Okay, cobalt +3 is low spin state here with having the configuration T2G6 EG0, and since it is low spin, so it is a diamagnetic in nature as well as EPR inactive. It does not contain any unpaired electron. Now you see here this R can be, that's why it is given as R here, so it can be substituted. If it is Cn here, R if equal to Cn, then it becomes cyanocobalamine. We call it as a cyanocobalamine. If R is no CN, there is no cyanide here, so simply it is called as cobalamine. Okay, if R is substituted by cyanide molecule here, then it is called as cyanocobalamine. So in normal cases, usually, so cyanide is been replaced by H2O, so water molecule, so it is called as hydroxycobalamine. If it is H2O, it is called as hydroxycobalamine, and if you substitute R by CH3, it is called methyl cobalamine. So I have given here, if it is CH3, it is called methyl cobalamine. And if the fifth position here, if R is equal to, that's what 5-deoxyadenosine ligand. So here, if R is if we substitute with deoxyadenosine ligand, it is called as coenzyme B12. Okay, so it depends on this. So now we are studying what? Vitamin B12 coenzyme. So when it becomes coenzyme, when the R is substituted by deoxyadenosine ligand. Remember this 5-deoxyadenosine ligand, 5-deoxyadenosine ligand. So if we substitute this, it becomes now coenzyme B12. We call it as a coenzyme B12, and see, it can be reduced now. So if vitamin B12 is reduced by 1 electron, it changes to vitamin B12 copper +2. From copper +3, it will change to copper +2. Yes, that is a labelization. So here metal to carbon labelization is very beautiful here. So metal to carbon. Okay, so in organometallic this one, so CO and C. So here vitamin B12 labelization is very good. So here one electron, if is been changed, if it reduced, it becomes vitamin B12 copper +2. So extra electron, where it will be placed, it will placed in dz square, and hence the extra electrons, it becomes a paramagnetic and EPR active. So if you reduce this by 1 electron, so it becomes paramagnetic as well as EPR, okay, electron paramagnetic resonance active. And further it can also be reduced here from cobalt +2 to cobalt +1, 1 electron reduction again we can do it. And this is called a super reduced form of the cobalamine. Now it becomes a super reduced form of the cobalamine. See, so that is a flexibility labelization, I can say, of the COC bond here, and that is also EPR active, and it will have 2 unpaired electron now, and this is brown in color. Okay, this is a brown in color, and why that is brown in color? Because of pi star transition in the corine ring. This is called as a corine ring, not porphyrin ring. I have told you the difference between corine and porphyrin ring. Just the difference is what? CH bond here. So pi pi star transition in corine ring. So this is brown in color, but the reduced form CO plus 2 is blue in color. Okay, this is what we can see change in the color. Okay, CO plus 2 is blue in color, but CO plus is brown in color. Okay, second reduced, so the super reduced form is a brown in color. Okay, now this is a water soluble. We know that this vitamin is a water soluble vitamin. Okay, so that's why it can be taken orally, because this vitamin is not synthesized by the bodies or plants, but it is synthesized in the bacterias. Okay, so human beings require this vitamin B12 to take orally. So we have in the market that B capsules and all, so that's a vitamin B12 capsules, and we take it orally, and it is not so harmful also. So we can take it without the advice of doctors, because it is a water soluble, and it can be, if it can be passed to the urine. Okay, but we should be very careful, not to take without the prescription of the doctors. Okay, like this. Then if what will be the deficiency? If the deficiency of cyanocobalamin or vitamin B12 is there, so it leads to pernicious anemia. Okay, it leads to pernicious anemia. So what is this pernicious anemia? It is nothing but low level RBCs. Okay, so RBCs will be very less in our body synthesis, and that leads to sometimes tiredness. Okay, depression. Okay, depression or confusion. So this type of deficiency, this type of disease, it will have. Okay, and as I said, it cannot be synthesized in the body and plants, but it can be synthesized in the bacteria. And where we get this vitamin B12, we get in meat. Okay, fish. Okay, egg and milk products. So in this we will get these are the sources of vitamin B12, and it was first isolated from the liver. Okay, it was first isolated from the liver, and this coenzyme act as a prosthetic group. Remember this, the coenzyme act as a prosthetic group. What is prosthetic group? That it can bind to several enzymes in the body to activate it. Okay, so this can bind to the several enzyme to carry on the function, and later on it can detach from the enzyme and it will bind to another enzyme. So that is called, it is a prosthetic group. So that's why we call it as a biological Grignard's reagent also. So that is what it will give. It will help in functioning of many enzymes to get many different products. Okay, so that's what it's also called as biological Grignard's reagent. Some of the functions and properties of this is isomerase reactions. It helps in the isomerase reaction, where we can see that it is called as 1,2 shift. Okay, isomerase enzyme. So this utilize this vitamin B12 in 1,2 shift here. So see, this is a molecule. So carbon 1, carbon 2 is there. So here hydrogen is there, H is there. So it will replace now here X in the 1 and 2 in the hydrogen. So replacement, simple replacement, 1,2 shift. So this isomerase enzyme, with the help of cyanocobalamin, that is vitamin B12 coenzyme, will help in such type of isomerase reaction in the body. Second, if you see diol dehydrase, again diol dehydrase is the enzyme which with the help of vitamin B12, so this is a diol dehydrase. Okay, CHOH. So you can see here geminal is formed here in the first case, with the help of vitamin B12. So this is going to form here. Then dehydration, you get aldehyde. See, from diol dehydrase is converted to aldehyde in the by the enzyme dehydrase in the presence of, with the presence of whom? Vitamin B12. So this is also important reaction happening in the body. Okay, conversion. Similarly, see here conversion of glutamic acid. Third, if we go glutamic acid to methyl aspartic acid. So this is done by glutamase, okay, mutase enzyme with the help of vitamin B12 coenzyme. So this is what happened. This is a glutamic acid, CHHCOH. So this is converted to methyl aspartic acid. CH3 is methyl, CHRCOH aspartic acid. This is called as methyl aspartic acid. So this is the conversion of glutamic acid to methyl aspartic acid, which is done by mutase, glutamic mutase enzyme with the help of vitamin B12. So this is how, these are some of the functions of the proper functions, or I can say, of the cyanocobalamin vitamin B12 enzyme, which will help for different enzymes to carry out the properties very fast and give the resulting products. And if you see here, this is only organometallic compound I said here. So for which is we require this in the body for proper functioning of the brain as well as nervous system. Okay, we require this for proper functioning of the brain and the nervous system. So this is what about the cyanocobalamin. Hope you understood this concept of vitamin B12 cyanocobalamin. Okay, so very important in biological term. In the next class we will go for the next concept of the bioinorganic, that is photosynthesis, PS1 and PS2 system. Okay, thank you very much. Have a nice day.